topic, cloning vector, and this is in the lesson Principles of Biotechnology. Now, starting with what actually is a vector. Now, vectors are specific carriers which carry specific pathogens and they transmit and cause infections. In this case, we are learning about plasmid, virus, and bacteriophage. Now, why plasmid is being used as a vector is because we can introduce our desired or our beneficial gene through a plasmid into a host cell, in this case E. coli, and therefore we can clone that E. coli into many desired cells. Right? Now, the main difference between plasmid and bacteriophage is their copy number. Plasmid has 2%, while bacteriophages, they are 100%. Therefore, bacteriophages, they are more of an advantage than plasmid, but in this topic, we will mostly be using plasmid as for learning. Now coming to the key components of vector, right? Now cloning vector, basically it contains all these elements such as the origin of replication, it contains these selectable markers, and they have these multiple cloning sites over here. Now, what is origin of replication? It's one of the salient features that the cloning vector offers, right? The origin of replication, or ORI for short form, it's a specific DNA sequence that enables the vector to replicate independently within a host or within a host bacterial cell, right? So, it is the site of initiation, site of linkage, or site of replication, right? Not only that, but the ORI is also responsible for maintaining the copy number of the plasmid, which can be high in the case of bacteriophage and which can be, low 2% in the case of plasmid. The next salient feature that we will be discussing is selectable markers. Now, selectable markers are these genes, these specific genes, which help us to identify between non-transformants and transformants. Now, what are non-transformants and transformants? Now, transformants are that cells which have successfully taken up the cloning vector, and non-transformants are vice versa. They have not taken up the cloning vector. Selectable markers are specifically used to identify these non-transformants and eliminate them because we don't need them, right? Now, in the cloning vector that we will be learning, which is the PBR322, the main two selectable markers that we are discussing is the ampicillin resistant gene and the tetracycline resistant gene. And preferably, only two selectable markers are being used in the cloning vector, and I'll explain to you why in a few minutes. Now, these are the various restriction sequences that we can see over here. In this topic of discussion, we'll only be using the BAM H1, but there are many restriction sequences, and we can ligate anywhere, insert DNA anywhere, but that will just make our process more complex, which we do not want. It will make it more chaotic, which again we do not want that. Next, coming to the cloning sites. Now, cloning sites is a specific region within the cloning vector where it contains these multiple restriction sequences, right? So these restriction sequences, like I said, like these are all of the restriction sequences. There are plenty. There is not one or two. There's multiple restriction sequences where we can ligate. But for this topic of discussion, we are only using the BAM H1. Now, when the foreign DNA is inserted or ligated at the BAM H1. For now, right? There are three types of cells which is being produced. One is the transformed recombinants, transformed non-recombinants, and non-transformed. Now, how do you know it's transformed recombinants? If the foreign DNA is ligated at the BAM H1 or your restriction sequence of your choice, right? At the site of tetracycline, these cells will develop insertional inactivation and will not grow in the tetracycline medium but ampicillin medium. Now, what does insertional inactivation means? Basically, when you insert a foreign DNA, an alien DNA into these genes, they will not function anymore. They will become inactive. That is what insertional inactivation means. So they will not respond to the tetracycline medium, but they will respond to the ampicillin medium, ampicillin resistant gene medium, right? Then we come to the transformed non-recombinants, these, they don't pick up the foreign DNA. So therefore, they are allowed to grow in both the mediums. They can choose any, they would grow in both ampicillin containing medium as well as tetracycline containing medium. Then lastly, we are coming up to the non-transformed. Here, they cannot, these cells, they cannot survive in any of the two mediums given. Now this process that I have just discussed to you, it's a pretty long procedure. It's very tiresome, very tedious, and also it involves simultaneously, playing with both the mediums, which, you need to be really patient to do that, right? So for that specific reason, scientists have developed an alternative method. Now, why this alternative method is more easier, more beneficial is because it does not have any selectable markers involved, nor restriction sequences, right? It's clearly ligated, your foreign DNA is clearly ligated in the E. coli cell itself. It can be directly inserted into the E. coli cell itself, right? Now, in the E. coli cell, there is this beta-galactosidase gene of the E. coli, and this serves as a cloning site, right? And this is being maintained in a chromogenic substrate medium, right? This chromogenic substrate medium contains a protein medium with colored compound chromophore. Now, I discussed about insertional inactivation. When we insert this DNA, our preferred foreign DNA, into this site, right, into this cloning site of beta-galactosidase gene, right, there are two possibilities that can happen. One, it experiences insertional inactivation, right, and when the beta-galactosidase gene, it's supposed to produce an enzyme called as beta-galactosidase, right? Due to insertional inactivation, it does not produce them. Therefore, it will produce colorless colonies in this chromogenic substrate, right? Now, if they have not picked up this foreign DNA, that means they are not experiencing insertional inactivation, they will be producing blue colonies. Now, if they are producing blue colonies, that means they are non-recombinant, and if they are producing colorless colonies, experiencing insertional inactivation, they are recombinant. Now, remember when I told you that there has to be two selectable markers present whenever we do the procedure, right? So, one selectable marker is used to identify between non-transformants and transformants, while the other selectable marker experiencing this insertional inactivation will help us to identify from non-recombinant to recombinant. So this is the colonies being produced. As you can see, these blue colonies, right, they are the non-recombinant. They have not picked up the foreign DNA, while these colorless colonies, they have picked up the foreign DNA. They do go under insertional inactivation. And this is also called as the blue and white screening process. Now coming to the final topic that I'll be discussing, which is vectors used to transfer gene into plants and animals. First up, we are coming with the Agrobacterium cumifaciens. It is a natural vector, right? It's a bacteria, natural vector, where it causes infection in plants. Now, how it causes infection is that it has this TI gene, which we call as the tumor-inducing gene. Now, this tumor-inducing gene, it causes infection in plants, right, by injecting its TI gene into the plant cell. And you know what happens? The plant cell, it will get to the DNA, it will start replicating, and then there is production of disease proteins, and there is multiplication of these disease proteins, and therefore they form tumors, or in this case we call them as gaps. Now, this is a threat to plants. Like I said, it's an infectious disease for the plants. But that can be prevented, and it can be used in a useful manner, a harmless manner, by, instead of using TIG, we can use our own desirable, our own target DNA by injecting it into the plant cell for us to express its own phenotype. Now coming to retrovirus, similarly it's called as the oncogenic virus, where the cancer-causing gene is being replaced by the gene of interest. We can replace the cancer, it's a sort of, we can call it also like a gene therapy, because we're all basically altering the DNA, right? And the cancer can be stopped, and we can replace it by our gene of interest and our target DNA. So is there any questions that you would like to ask? Yeah, yes.
Why is the selection of recombinants due to insertional inactivation of beta-galactosidase preferred over other antibiotics?
Like I said, the normal cloning vector procedure that I just discussed, it's a very tiresome process. The other process, the alternative method, the beta-galactosidase method, it does not require any selectable markers or restriction sequences, and the process is much simpler and less complex compared to the other one. Yeah?
You have explained that, in your explanation you have given that the foreign DNA was inserted into the tetracycline site. What will happen if the foreign DNA was inserted into the other site, that is ampicillin site?
Vice versa, and exactly the opposite. That means it won't grow in the ampicillin medium, but it will grow in the tetracycline medium. Exactly opposite. Anything else?
Why does the vector need to have very few, or probably a single recognition site for the restriction enzymes?
There are many of those sites present in the cloning vector, and they have their own sequence, those nitrogenous bases, palindromic sequence that we just discussed, right? It's possible, it's very much possible to ligate and insert in all of those sites, but it will make the process really tiresome. It will make it all chaotic. So we usually just go for one or two. Is that all?